So today's lecture will be the first lecture in a 5-lecture series all about enzymes, which are proteins that catalyze chemical reactions necessary to biological systems. So today's lecture will just be an introductory lecture on enzymes, and we'll establish how enzymes are able to catalyze biochemical reactions. So we'll start with a quick discussion on thermodynamics of chemical reactions, including a discussion of Gibbs free energy. Then we'll talk about how enzymes are able to increase the reaction rate of these chemical reactions. And then we'll end with talking about how enzymes are able to interact with substrates and how this leads to that increased reaction rate in these biological reactions. So let's get started. Lecture number 7, enzymes.
Right. So to put it in the most simple terms possible, enzymes are biological catalysts. So a catalyst is a molecule that accelerates the rate of a chemical reaction. And so enzymes are proteins that catalyze biochemical reactions at a rate that is conducive to sustaining life. So you have billions and billions of reactions going on at any particular moment in your body at any particular time. So those reactions would not occur at a rate that could sustain life. So what enzymes do is they speed up the rate of those reactions to a rate that is conducive to sustaining life and all the biological processes and cellular processes that occur in your body. So here's a list of some different enzymes, and it shows the different rates. Basically how enzymes speed up the rates of these reactions. So, for instance, here we have an enzyme OMP decarboxylase. So the reaction that this enzyme catalyzes the half life without the enzyme would be 78 million years. But OMP, when you add OMP decarboxylase, this is able to occur about 39 times per second. So obviously, it increases the rate of this reaction to a very big extent. And so here we have some other examples of different enzymes and how they speed up the reactions that would normally take seconds to hours to years to millions of years. They can now occur many, many times in a second. So, a good example and one of the fastest enzymes that is of known is carbonic anhydrase. So, our carbonic anhydrase catalyzes the reaction of CO2 plus H2O, and it converts this to carbonic acid. So, H2CO3. And so carbonic anhydrase increases the reaction rate of this reaction from less than one reaction per second to about a million reactions per second. And so this is very important to sustaining life, and it sort of illustrates how important enzymes are and also their activity, which is to act as biological catalysts that increase reaction rates of chemical reactions in your body to a rate that is conducive to sustaining life. Now, another feature of enzymes that's very important is that enzymes are highly specific. So they have high level of specificity for the type of reaction that they catalyze and the substrates that are involved in that reaction. So an example that we'll come back to over and over again are proteases. So proteases are specific enzymes that cleave peptide bonds between amino acids. So we've talked about how proteins are made up of amino acid polymers formed by peptide bonds. Proteases actually cleave that peptide bond using a hydrolysis reaction. So the peptide plus water results in the breaking of that peptide bond into a carboxyl component and an amino component. So this reaction would take hundreds of years without the activity of proteases. So proteins or peptide bonds are quite stable. They can be set around, be there for 1,000 years, but it happens in just milliseconds with the activity of proteases. So proteases are very specific. They only cleave peptide bonds. They don't cleave lipid bonds. They don't cleave carbohydrate bonds, for example. So Enzymes are highly specific for the substrates that they interact with and the types of reactions that they catalyze. And this specificity goes even further. So different proteases don't only just cleave peptide bonds, but specific proteases will only cleave specific types of peptide bonds. So they are highly specific. So some examples of this are the protease trypsin. So trypsin will hydrolyze peptide bonds only after a lysine or an arginine residue. So it cleaves this bond, this peptide bond right here, but only if there's a lysine or an arginine residue directly before that peptide bond. In contrast to this, thrombin, which is also a protease in the same class as trypsin, will only cleave peptide bonds between an arginine and a glycine residue. So this is highly specific and is how your body is able to function. And this specificity is dictated by the three-dimensional structure of each of these enzymes. So trypsin and thrombin have a lot of features in common, including the residues that catalyze this reaction. However, they have slight or major differences in the three-dimensional structure of each enzyme that dictates this highly specific activity. So people have been studying enzymes for quite a while now, and it's been determined that all known enzymes can basically be categorized into 6 different groups or 6 different classes. And they're actually numbered 1 through 6. So number 1 are oxidoreductases. So these are enzymes that transfer electrons between molecules. So they catalyze redox reactions or oxidation reduction reactions. So they'll oxidize or reduce different molecules. Number 2 are transferases. So these are enzymes that transfer functional groups between molecules. So from one molecule to another. There are hydrolases, so that's number 3, hydrolases. So these enzymes cleave molecules by the addition of water. So that's the example that we just saw with proteases. So proteases cleave peptide bonds by the addition of water, so they are considered a hydrolase. There's also lyases. So lyases add or remove atoms or functional groups to remove or form double bonds in molecules. Isomerases, that's number 5. So isomerases move functional groups within a molecule. So they convert one molecule into an isomer of that same molecule by moving a functional group within the molecule. And then number 6, the last are ligases. So ligases actually ligate molecules. So they join 2 molecules together in a reaction powered by ATP hydrolysis. So actually the formation of those peptide bonds would require ATP hydrolysis and be performed by a ligase as the type of enzyme that performs that activity. So there's actually these 6 classes you can use this to give numerical designations for each molecule, starting with these classification. So for example, trypsin, which is a hydrolase, is designated among all known enzymes as 3.4.21. And so the 3 starts with the hydrolase, and then the 4 and the 21 are based on other features of trypsin. So there's 6 different classes of enzymes. All known enzymes can be categorized into these 6 different classes. And so another feature of enzymes that comes up over and over again is that many enzymes use cofactors for their activity. So these are the small molecules that we call cofactors that enzymes use to actually catalyze the reactions that they catalyze. And so there's basically 2 different types of cofactors that can be categorized into 2 main different groups. So the first one are coenzymes. So these are small organic molecules that are derived from vitamins. And so these coenzymes can either be very tightly bound to the enzyme, basically always be with that enzyme, and that would be called a prosthetic group, or they could be loosely bound. And then it's sort of called a co-substrate because it almost acts like another substrate with the substrate of the end of the reaction that they're catalyzing. So coenzymes are one type of cofactor, and then metals are the other type of cofactor. So those are commonly found in all sorts of different proteins that do all sorts of different activities. And so here we have a list over here of different cofactors. So and that the enzymes that some of the enzymes, an example of an enzyme that uses each of them. So thiamine pyrophosphate, FA, which is called TPP, FAD, NAD, PLP, coenzyme A, biotin. So these are very common. They're derived from vitamins and a lot of times vitamin deficiencies. So some of the diseases that a vitamin deficiency would cause is because you don't have these cofactors to catalyze a specific reaction. So an example would be beriberi, which is a disease from a lack of a beta vitamin that contains TPP, this thiamine pyrophosphate, and that causes metabolic dysfunction because pyruvate dehydrogenase is very important in metabolism. So the other ones are metals. So there's lots of different metals that we need. These can also be, I guess they would be considered nutrients rather than vitamins. But if you take a multivitamin, it's going to contain some of these small metals because you need them in small amounts for certain enzymes to work. So including zinc, which is used by carbonic anhydrase, which we talked about earlier, manganese, nickel, molybdenum, selenium, no, this is manganese. That's magnesium up here, and then potassium. So the metals are commonly used in all sorts of different enzymes. And so the term for an enzyme that contains its cofactor would be a holoenzyme. So you can just think of the enzyme as whole at that position. And then an apoenzyme. Apo means without. So that would be an enzyme that does not contain its cofactor. So cofactors are very important for enzyme activity, and they're commonly used by a lot of different types of enzymes. All right, so now we can actually get into how enzymes work and how enzymes influence the reaction thermodynamics. So we'll have a discussion on Gibbs free energy and reaction thermodynamics. So Gibbs free energy, which is referred to as G, capital G, is the measure of energy that is capable of doing work. So when we're thinking about a reaction, we have products, reactants, and the products, so the chemicals that we start with, and then a chemical that we end up with. And so the Gibbs free energy would be the amount of energy that's capable of doing work within that molecule. So a change in Gibbs free energy is going to be called delta G, and we'll discuss that quite a bit today. So delta G provides information about the spontaneity of a reaction. So whether a reaction would occur spontaneously without any external input. But it doesn't tell us about the rate. So it doesn't tell us how fast a reaction will occur. So if there's if the free energy of the system, if there's a negative delta G, so basically you're releasing energy, that means that the reaction is going to take place spontaneously. But that spontaneously doesn't mean that it's going to happen automatically. As we saw from that one of those first slides, that reaction could be spontaneous, but it could take 78 million years to occur. So delta G tells us whether the reaction is spontaneous, but not its rate, not how fast a reaction will occur. So enzymes, and this is very important, enzymes do not change the delta G of a reaction. So enzymes don't change how much energy that is capable of doing work is in the reactants or the products of a reaction. Enzymes don't change the delta G of a reaction. That's very important. What enzymes do is they change the rate of the reaction. So they can take a reaction. They're not going to change how spontaneous it is, but they can take it from happening in 78 million years to just a few seconds. So enzymes do not change the delta G of a reaction, but enzymes do change the rate of a reaction. So hopefully this will make a little bit more sense now that we can actually look at a graph of what I'm talking about. So here we have basically the progression of a reaction. So we have reactants A and B, and those are going to be converted into products C and D. So over here on the y-axis, we have the free energy of this of both this reaction, so the reactants and the products, and then on the x-axis we have the reaction progress. So in this particular case, you can see that the reactants A and B have a higher Gibbs free energy than the products C and D. So this reaction is going to have a negative delta G because we're having lower free energy at the end than we're going to have in the beginning. So when the delta G is negative, the reaction is going to be spontaneous and it's going to be exergonic. So it's going to release energy. So if you have higher energy in the reactants and then you have lower energy in the products, that difference in Gibbs free energy is going to be the delta G. And here it's going to be negative, and that energy is going to have to go somewhere, so it's going to be released. So when you have a negative delta G, as in the case here, that reaction will both be spontaneous and it will be exergonic, so it will release energy into the system. Now here we have the exact opposite case. So in this reaction, the reactants A and B have a lower Gibbs free energy than the products C and D. So that means this delta G, the difference in Gibbs free energy, is going to be positive. So this reaction is not going to occur spontaneously, and it's going to be endergonic. So it requires an input of energy. That energy that's going to make up that positive delta G has to come from somewhere. And usually when we're talking about enzyme reactions, that's going to be in the form of something like ATP hydrolysis. But we'll get into that a bit more later. But so it's important to understand that in a system like this, where the delta G is positive, the reaction is not spontaneous and it's endergonic. So it requires an input of energy for the reaction to occur. Now, the last example would be where delta G is 0. And so this would be when a reaction is at equilibrium. So there's no change in Gibbs free energy between the reactants and the products. So there's not going to be any change in the amount or the free energy in the reactants and the products. So the reaction is going to be said to be at equilibrium. So I mentioned it before, but it's very important. I want to reemphasize it, is that delta G does not describe the reaction rate, so the speed at which the reaction will occur. It also doesn't describe at all the pathway, so how the reactants are going to be converted into products. So delta G only describes the free energy difference between the reactants and the products. So it'll tell us whether a reaction is going to be spontaneous or not spontaneous, whether it's going to be exergonic and release energy or endergonic or require the input of energy. But it's not going to tell us how fast that reaction will occur or the pathway. So both these reactions here have the same delta G, even though this transition state, so the state where this reaction is neither fully reactants nor fully products, this reaction has one transition state. This reaction here has 3 different transition states, but both reactions have the same delta G. And also, importantly, I keep saying this, but delta G does not provide information about the reaction rate. So why I keep emphasizing this is because enzymes the point of enzymes, or the function of enzymes, is that they increase the rate of reactions. So enzymes cannot alter the reaction equilibrium, but they can change the reaction rate. So this is demonstrated in this graph here. So over here we have the formation of the reaction product, and at the bottom is time. So when no enzyme is present, this reaction is going to occur very, very slowly and in a linear way. So it's going to keep going and keep going over seconds. And then eventually at hours after, it'll get to this level up here. So enzymes don't change how the reaction would normally go. They just make it go a lot faster. So now this reaction that would normally take hours to get to this level, that happens in seconds when you add an enzyme to the system. So enzymes don't alter the reaction equilibrium or the delta G, but they do change the rate of a particular reaction. So how do enzymes do this? How do enzymes increase the rate of a particular reaction? So how enzymes do this is they facilitate the formation of that transition state that I mentioned briefly before. So reactants, or what would be called substrate, an enzyme substrate, they proceed through a transition state before they convert to products. You can see here, and this transition state is going to be at a higher energy delta G level, and it's indicated by this X double cross here. So the reactants will convert into the transition state before they're going to convert into products. And that transition state, again, is this X double cross. So the activation energy is this energy that you have to put into the system for the substrate or the reactants to form that transition state. So the delta G double cross is the activation energy. So there's got to be some sort of energy that's put into a system to allow the reactant to form that transition state before it converts into products. Now, how enzymes work is that they lower the activation enzyme by stabilizing that transition state. So normally that transition state may not be very stable. What enzymes do is they stabilize that transition state, and then you can see here that it greatly lowers the activation energy of this reaction and allows it to go at a higher rate. So this is how enzymes increase the rate of a reaction. They stabilize the transition state by lowering the activation energy required to form that transition state. And that's how you can take a reaction that takes 78 million years to do it in a few seconds, because it needs to form that transition state. And that 78 years, that results from the inability for that reaction to quickly move into the transition state. So again, enzymes increase the rate of reaction by stabilizing the transition state and lowering the activation energy required to form that transition state. All right, so now that we've gone through the thermodynamics and how enzymes influence the thermodynamics, we can start looking at some other features of enzyme activity. So one feature that you need to know is that enzymes bind the substrate at what's called an active site. So the first step in enzymatic catalysis is the formation of an enzyme substrate complex. So you're going to have enzyme plus substrate, and that's going to convert into an enzyme substrate complex. And that can go back to enzyme and substrate, but it can also convert into enzyme plus product. So that enzyme substrate complex is going to happen because enzymes will bind substrate at a cleft called the active site that includes residues that bind that particular substrate and perform catalysis. So the active site has two jobs basically, to bind that substrate and to perform the catalytic reaction. So one of the features of active sites, so if you think about the tertiary structure of proteins, a lot of times active sites are formed by residues that are located far away in the primary sequence of the protein. But once the protein folds into that tertiary structure, those residues can be located quite near to each other, and they can form a cleft that can bind that substrate, here as shown in red, at the active site. So this is one of my favorite proteins, LSF2, and you can see there's this cleft here where it binds a carbohydrate substrate directly over the site where catalysis takes place. So enzymes bind substrate at the active site. The active site is important, is two jobs. It binds substrate and it performs catalysis. So now we can look a little bit closer at actually how enzymes bind substrates at their active site. And to help us understand this, there's two different models that we'll cover. that describe how enzymes bind substrate at the active site. So, the first model, which is actually a little outdated, um, which describes the enzyme substrate interaction, is the lock and key model. So, in this model, if you think about it, this sort of substrate would have a very specific chemical characteristics and a very specific shape. And that's going to correspond precisely with the chemical composition and the shape at the active site. So if you think that's why it's called the lock and key model. So there's a keyhole and a key, and they're sort of designed to fit perfectly to form the enzyme substrate complex. Now, the more this is a good understanding for, a good model to understand generally how enzymes work. The substrate corresponds to the shape and the chemical characteristics of the active site, allowing it to bind. But as we've gained more and more information on different enzymes, more about their structure, more about the enzyme substrate interaction, a more accepted model now is the induced fit molecule. So in this model, the enzyme actually changes shape upon substrate binding to accommodate the substrate transition state. So we already went over that the enzymes act by accommodating the transition state and lowering that activation energy. So the best way to do that would have an active site that sort of corresponds to the substrate, but actually changes conformation when the substrate binds so that it can accommodate this substrate transition state. So the substrate is not going to be floating around in that transition state. You need it to bind to the enzyme and then the enzyme to be perfectly accommodated mostly to that transition state. So that's how this induced fit model works. So the substrate binds, that causes a structural change in the enzyme, and that allows the enzyme to accommodate the transition state. So, and I just want to point out, so both of these models describe how substrates are bound to enzymes by multiple weak interactions at the active site. So there's going to be a lot of different residues for the most part that are going to be involved in this enzyme substrate complex, and that the structure of the active site is most complementary to the transition state. That's what's going to make the most efficient, the best enzyme. And this induced fit model here is describes that the best, and it's most, highly supported by the evidence that we have. And so, how does this binding or this formation of this enzyme substrate complex actually lower the activation energy required for the substrate to form the transition state before being turned into products? So the answer to this is binding energy. So binding energy is the free energy that's released when an enzyme interacts with substrate. So that enzyme substrate interaction, those weak interactions between the substrate and the amino acids or the cofactor on the enzyme are actually going to release energy. And that results in a lowering of the delta G double cross, which is the activation energy. So this binding energy effectively lowers the activation energy of the reaction, and this binding energy is going to be the greatest when the enzyme interacts with the transition state. And so that fits most well with our induced fit model of enzyme substrate interaction. So again, enzymes bind substrate, which lowers the activation energy, which highly increases the rate of a reaction, and the energy to lower this activation energy comes from the binding energy of the interaction between the enzyme and the substrate. And so, one of the best, a big piece of evidence that enzymes are that they work best when stabilizing the transition state is that inhibitors of enzymes often resemble the transition state of a substrate. So transition state analogs are molecules that resemble the transition state in an enzyme substrate, and they can often bind with a lot higher affinity than the natural substrate. So an example of this would be this molecule pyrrole 2 carboxylic acid, which is a transition state analog for the reaction catalyzed by proline racemase. So proline racemase takes L-proline, which is an isomer that's shown here, and then it converts it into D-proline, which is the chiral isomer of L-proline. So for proline racemase to convert L-proline into D-proline, it has to have this planar transition state where this carboxylic acid group and this hydrogen group, or this carboxylic acid group, basically goes from pointing towards us to away from us, or this hydrogen does. Anyway, so it has to go through this planar transition state. So this molecule, pyrrole 2 carboxylic acid, is designed to mimic more that planar transition state than the L or the D proline isomers. And so this is a very a very effective inhibitor of proline racemase. So this is just more sort of information reiterating that enzymes work best when they stabilize the transition state of a reaction, so inhibitors often resemble the transition state.
All right, so that concludes the first of the 5 lectures that we're going to have on enzymes, and there's a few points that I'd like to emphasize. So first is that enzymes catalyze biochemical reactions, which means they increase the rate of chemical reactions to a level conducive to sustaining life. You also need to understand that enzymes are incredibly specific when it comes to their substrate preferences. So enzymes bind the substrates at their active site, and the enzymes often use cofactors at their active site to help catalyze these reactions. You also need to understand the basics of thermodynamics of these chemical reactions. So you need to understand the concept of a change in Gibbs free energy, or delta G. You need to know that exergonic reactions mean there's a negative delta G and there will be a release of energy, whereas endergonic reactions have a positive delta G and they require an input of energy. So you also need to understand the concept of a transition state and activation energy, which is the energy required to form this transition state so the reaction can go from substrate to products. And importantly, you also need to understand that enzymes enhance the rate of reactions by lowering this activation energy because they bind the substrate preferentially and they help form the transition state. So we'll get into more details in this in the next lecture, and if you have any questions on this, please let me know.